Hi, my name is Nina Jorgensen. I am a third year studying biotechnology at UC Davis, and it is my pleasure to be presenting to you about the use of CRISPR-Cas9 to create genetic knockouts that may increase meiotic recombination in lettuce. And just a heads up, I'm going to turn off my camera so that when I'm presenting and zooming in, you won't be having my face cover anything during the presentation. To start, maintaining genetic diversity in living organisms relies heavily on the shuffling of genetic traits from parents to their offspring during meiosis. The crossover events that actually drive genetic diversity are relatively rare, with only about 1 to 3 crossovers per chromosome, but we've recently discovered 3 anti-crossover genes in plants, known as FANCM, FIGL1, and RECQ4, that may be responsible for restricting the rate of crossover events during meiotic recombination. Using genome editing tools has become an increasingly attractive strategy to knock out these anti-crossover genes, with the potential to enhance crossover rates genome-wide. Increasing the rate of crossover events in plants may allow plant breeders to unlock previously inaccessible traits and develop more efficient breeding programs that could tackle environmental stressors due to climate change, such as drought or extreme temperatures. So to get started with our research, we wanted to target these anti-crossover genes, and you can see here that in each gene, there are 3 to 4 sites that we decided to target for editing. You can also note that each site is in an exon. Exons are the protein-coding parts of genes, so by targeting and knocking out an exon, we would be able to knock out the function of the entire gene. When we're preparing to knock out a gene, we have a few questions in mind. 1, were we able to successfully edit and knock out the gene? 2, if so, did this knockout affect rates of crossover? And 3, will this knockout be passed down through generations? If this knockout is passed down and we know that it does affect crossover rates, then this technology can be especially helpful for plant breeders that are interested in lines of plants and seeds that have higher rates of meiotic recombination. So here's a general overview of how we induce knockouts in plants. Let's focus all the way on the left here, starting with our plasmids and our PCR products. A plasmid is just a piece of circular DNA that is independent of a cell's chromosomal DNA. You can see over here with the agrobacterium, the plasmid and the chromosomal DNA. In my research, I built 3 different plasmids that I call level zeros or L0s. One L0 has a promoter sequence, another L0 has sequences encoding for the CSY4 protein and the Cas9 protein, and a third L0 has the terminator sequence. These L0s were combined to build a level 1 plasmid, and now the promoter sequence can recruit an RNA polymerase to read through the entire L1, translate the CSY4 and Cas9 sequences, and then end at the terminator. To make use of the CSY4 and Cas9 proteins, we'll need another L1 that encodes for CSY4 repeats and guide RNAs. The CSY4 protein will cut out the repeats, and the guide RNAs will guide Cas9 to different parts of the genome. I built the second L1 using PCR products rather than plasmids. The PCR products are just fragments of DNA as shown here on the left, and we have all these different fragments encoding for a promoter, CSY4 repeats, guide RNAs, and a terminator. We can use all of these PCR products to build our guide RNA array, and then we can use both L1s to make the level 2 plasmid that goes into our plants. This level 2 also contains the DS-red gene and a kanamycin resistance gene. The DS-red allows the plant to fluoresce red under UV light, and the kanamycin resistance is mostly useful before plant transformation. After transcribing and translating the DNA in this level 2 plasmid, the plant cell should be able to use the CSY4 proteins to cut at the repeats, and the resulting guide RNAs should be able to guide the Cas9 protein to our target sites. The Cas9 is going to be able to make cuts in the DNA and induce editing. To see if our level 2 plasmid actually worked and induced editing, we use agrobacterium to transform the plasmid into a plant cotyledon. The cotyledon grows into a full plant, the plant produces seeds, we extract DNA from the plant seeds, or even a plant's leaf, and we send that DNA to a sequencing facility so that we can analyze sequences and see if and where editing occurred. So that's the full rundown. Make a level 1, then a level 2, then transform into plants, and analyze. So keeping all that in mind, we can take a look at what I did with the RECQ4B gene. This picture is the full level 2 plasmid that was transformed into plants. You can see here that I have the DS-red gene for the plant to fluoresce red, then the guide RNA array made from the PCR products, the CSY4 and the Cas9 sequences that encode for proteins, and the kanamycin resistance. You can also see in the guide RNA array that the CSY4 repeats are between each guide and that each of the 4 guides will target their respective sites on chromosome 9. So right now, I'm just waiting for my transgenic plants to grow. I'm somewhere in here, so analysis of the RecQ4b construct hasn't begun. That being said, I have been able to extract and analyze DNA from other transgenic plants that have similar constructs but target different anti-crossover genes. These transgenics are from Beth, who assigned and helped me with my project, and she was able to create constructs that target RecQ4a, FANCM, and FIGL1. We used both Sanger sequencing and amplicon sequencing to look at our transgenic DNA samples. Starting with RecQ4a Sanger sequences, there have been lines with editing, as seen in line 775 for site 1 and line 799 for site 4. Both lines have multiple alleles, multiple forms of the gene, which means editing did occur. Sanger sequencing isn't quite specific enough to tell which nucleotide is at the multiple allele positions, hence the Ks and Ys and Ms, but we know that editing did occur. Similarly, there have been deletions in other RecQ4a transgenic lines, such as the line 791 in site 1. These deletions shift the entire coding sequence backwards by 2 base pairs, effectively changing the protein sequence and structure and knocking out the gene's function. Not to mention, this edit was also found in the first transgenic plant, this T1, and then it was found again in the seed pool, the T2, so we know that these edits can be passed on to progeny. I also want to point out why the edits are occurring where they are. The Cas9 protein cuts just about 3 base pairs above the protospacer adjacent motif, which we call the PAM site. The PAM site is just the nucleotide sequence TTG, and if you look at our Sanger sequences, you can see the PAM site right here, and then the cut site approximately where the guide is, and then mutations. Now, moving on to our FANCM amplicon sequences, we noticed a deletion in site 2 and 2 insertions in site 3. This actually does make sense because site 3 had 2 guides, just in slightly different areas, hence the 2 different insertions. And unfortunately, we didn't actually find any lines with editing when we targeted FIGL1. With all this said, it's safe to say that editing is present, but it is also low efficiency. Our constructs were successful, but they may not have been the best for lettuce. And heritability was shown in our RecQ4a lines. Just before we move on, I want to point out this table. It shows the genes that we've screened so far, the number of lines we have, the number of sites we've screened in the genes, the number of lines that we've actually had with editing at a site, our percent of lines with editing, which I think is very important and kind of demonstrates the editing that is successful but maybe not the best for lettuce, and the heritability of our mutations. Even with what we know now, there's still so much to be done. I plan on screening my transgenic plants when they grow and produce seeds, and I'm currently working on cloning a new construct that we hope will have higher editing efficiency. If we look back to our Golden Gate Diagrams B section, you'll notice that instead of PCR products, we use many level 0 plasmids for our guide RNA array. That, and we no longer have any of the CSY4 repeats, nor the CSY4 protein encoding sequence. The guides will be cut out using the cell's natural machinery that will recognize the tRNA sequences, and it'll leave the guide sequences available for Cas9 to bind with. This guide tRNA array hasn't been tried in lettuce, so I'm super excited to see how it fares in the coming months. So just before I head out, I want to thank Lean, who had worked in the Michelmore Lab, for creating the CRISPR editing construct design that we were able to use for the rest of our anti-crossover genes. And I just want to thank you so much for coming and listening to me talk about my poster and my research. It's really fun, and I hope you enjoyed. Please feel free to leave any questions or comments or concerns that you have in the Go React commenters box, and I will get back to you as soon as I can. Thank you again.